Hi, I'm Erico Freitas, a postdoc researcher at the Center for Bioengineering of Invasive Species of Hydroelectric Power Plants. And in this presentation, I'm going to talk about the golden mussel filter feeding. My intention is to highlight the mechanism of particles capture and selection. So here we go. So I'd like to start with the mussel anatomy. So I brought some short videos of 3D reconstructions of the blue mussel, the Mytilus adulis, showing its basic anatomy. Although they are quite large compared to the golden mussels, so their anatomy might be quite similar. So at left, we see the bivalve. So its body is surrounded by two valves that are made up of calcium carbonate. So it's got three types of calcium carbonate, three polymorphs in the amorphous phase, and then a calcite, that's a trigonal phase, hexagonal trigonal phase, and also aragonite, that's an orthorhombic phase. At the middle we see, underneath the valves, we see the body of the mussel. So it's covered by the mantle, and inside the mantle we have what is our focus, the gills of the mussel. The gills are large structures compared to the other organs. So we see here the gills, they are actually double lamellar structures. There are two pairs, there are two double lamellar structures, a pairs of demibranchs. Its structure is it comprises of a series of parallel filaments, ciliated filaments, we are going to see in the next slides. So here we see, we have a closer view of that double lamellar structure. So the gills are very important because they are responsible for breathing and feeding. And also, just a minute. And also they are in direct contact with the environment. So that is important because they can serve as xenobiotic biotransformation. For example, antioxidant response, we can study an antioxidant response, signal transduction, and also innate immune response, and also osmoregulation, and also they can serve as environmental indicators. So we think that understanding of the golden mussel morphology, the Limnoperma fortnii, is the very step towards the establishment of strategies to control these invasive species. So we have here some light microscopy images that Amanda Moreira, a master student that I'm co-supervising, is working with. We see here at the picture above a cross-section view of the many filaments that comprise the gills. I think it's perpendicular to the chytnidial axis, and as in a closer view in that region here, so we see a set of pairs of individual filaments in a cross-section view, where we can see the frontal cilia and the lateral frontal cilia at the margin of each individual filament. All right, we see a longitudinal view, longitudinal section of one filament showing its W shape, and also we can see here the marginal food groove. The lateral cilia, as we actually you don't see the lateral cilia here in those images, but it is depicted in that animation. So here's a section, in that animation we see a section of one filament. So the lateral cilia are responsible for the water pumping. So as they beat the water pumping, the gills pump waters from the mental cavity towards the suprabranchial cavity. In this animation, in a vertical direction from the top to the bottom. And the lateral frontal cilia, as we see here, also in the picture and in this animation, they are responsible for the particles capture. They also transfer the captured particles to the frontal surface of the upfrontal surface of the filament, and the frontal cilia that are located at the upfrontal surface of each individual filament. They also transfer the particles to the marginal food groove, or maybe the water current, the frontal. It's not yet clear whether the particles are transported by the movement of the frontal cilia itself, or by the water current that movement produces. So there are two principles of particles capture mechanisms according to the literature. So they are based whether the bivalve has lateral frontal cilia or not. So one is called Cilia Trapping. So of course in bivalves with lateral frontal cilia, so where particles are separated to the main water current, and then they are transferred to the frontal surface of the gill filament by the action of the lateral frontal cilia. So that particle retention efficiency is also size dependent. So particles larger than let's say 4 or 5 micrometers, they are transferred by the lateral frontal cilia to the surface of the upfrontal surface of the filament. But smaller particles can either follow the flow of the water current around the lateral frontal cilia, or maybe they are driven by the water flow. Yeah, in bivalves without lateral frontal cilia, that mechanism is still poorly understood. So the literature suggests, according to the literature, it is suggested that mechanism is dependent on water flow rather than direct filtration and transport by cilia. So in that picture we can have a look at the real a live video produced by J. Evan Ward through endoscope, and we can clearly see some particles that are driven by the main water current into the mental cavity, and then they are stopped at the surface of the gills, and then they are captured and driven by the action of the cilia. So that's quite interesting to see that, because those are amazing animals, and the way they filter and capture particles, and then the way they select or reject the captured particles is important, so we can find better strategies to control that invasive species. And yeah, just to summarize, the particles are captured and the transport mechanism in filter feeding. They are almost serially dependent. And as we've already seen, the lateral frontal cilia transfer particles from the main water current to the upper frontal side of the gill filaments. And the frontal cilia, they are responsible to transport the captured particles towards the marginal food groove by the action of or maybe by their movement, or maybe by the metachronal wave their movement produce, as is depicted in that picture above. So the way the frontal cilia beat, so as they bend in a metachronal rhythm, so they produce the appearance of a wave, and maybe the water current close to the lateral frontal cilia, they are responsible to drive the captured particles towards the marginal food groove. That particles will then pass directly to the mouth after being sorted by the labial palps. The present understanding of the Mussel filter. So we know the particles are transferred to the frontal side of the gill filaments, or those particles are transferred by the lateral frontal cilia, or by the current they generate as they beat against the water through an angle of 90 degrees. And the frontal cilia then transport those particles towards the marginal food groove. So that efficiency is size dependent. So, and smaller particles are not, the efficiency is better for larger particles, larger than 4 micrometer size. And the smaller ones, they, as I told you, they follow the flow around the lateral frontal cilia, or are stopped by the lateral frontal cilia's branching cilia. But the particles rejection or ingestion, it's based on physical chemical interactions that can be sensed in the labial palps. Yeah, so it's important to understand that mechanism, because if you at hydroelectric power plants, they use chemicals to try to control the population of golden mussel. But it's important to us to understand how the mussel select or reject those particles, or which effects these chemicals cause to the gills and other organs. Yeah, so let's go ahead. So the understanding of particle selection by mussels is not fully resolved. There are some gaps in the literature. So we know it may depend on particle size, shape, surface properties, which alter their ingestion or rejection. So it's also stated that rejected particles are bound to cohesive mucus and deposited in specific place in the mantle cavity, and then transported by the cilia for their expulsion of pseudofeces. And interestingly, mucus covered feeding organs, like in the labial palps, they can mediate particle selection. So our question is, does the mucus in the bivalve gill filaments, the mucus produced in the gill's epithelium, do they play a role in the particles capture mechanism itself? So we tried to tackle that question in a work submitted to BMC Zoology. So that paper is not yet published, is in review, but you can download it and make your comments if you like. So it's still in review. So I'm going to show some highlights and some data. So we found in golden mussel highly organized and large amount of ciliary structures, also a highly energetic apparatus composed of we saw large amount of mitochondria, smooth endoplasmic reticulum, and glycogen granules in epithelial cells. Those energetic apparatus, they can be related to the morphofunctional structure of the cilia. And we also observed mucus-containing vesicles that are the leaf to the apical epithelium, and we think they may play a role in the selection and or rejection of captured particles in suspended water. So in that work we did a high resolution characterization of the outer structure of the gill's epithelium, and our goal was to better understand the morphophysiology aspects of the organelles and these structures, and we think that is the most important. We think it's important to highlight the mechanisms that of the particle selection and rejection. So we think the understanding of the golden mussel morphology might be the first step towards, as I told you, the establishment of strategies to control these invasive species. And to our knowledge, that is the first time high-resolution ultrastructure of the golden mussel being reported. So here we have some scanning electron microscopy secondary electron images, SEM secondary images of the gills of the golden mussel. So we see in image A, we see clearly the filament that juxtaposed filaments bound to an X that is called Cnidia X, and actually that's a demi-branch we see here above the inner demi-branch and underneath the outer demi-branch. In image B we see a closer view of that region where we can clearly see the ciliated filaments at its margin, and in the middle we see the marginal food groove, and you see a longitudinal view of one of these filaments showing its double shape. We can also see here some ciliary discs that cross-link each individual filament. So those pictures at the right depict those ciliated structures. Here you can have a closer view of an individual filament. In image A, we have a set of filaments. We can clearly see the ciliary discs. B is a high magnification image of that ciliary disc. As I told you, they cross-link each individual filament, and at the margin of each individual filament we see the lateral frontal cilia and the lateral cilia. Image C and D, they show in high resolution, in higher magnification, this lateral frontal cilia, and also some captured particles attached to the lateral frontal cilia. So in D it might be so you have a larger than 4 micrometer in diameter. It might be maybe an algae cell. And our transmission electron microscopy images, they revealed in high resolution the organelles we found at the epithelium of the gills. And in image A we see, we can clearly see the lateral frontal cilia in a stiffer position, and also some frontal cilia that protrude from the epithelial cells. So B is a higher magnification image of nearly the same area, where we can see the nucleus, the Golgi complex, some mitochondria, and some vesicles. They are sent to the apical region of the epithelium. So image C is also, we can also see the lateral frontal cilia, but in a cross-section view, which means they are bent in relation to the frontal surface of the gills. And we can clearly see sharper mitochondria. There are more mitochondria when we found out more sharper mitochondria easily seen when the lateral frontal cilia are bent, as we can see here in a cross-section view, than when they are stiffer in image A and B, for example. And this is a closer view of the apical region of the filament, where we can see also mitochondria, smooth reticulum, and lots of glycogen granules, and lots of mucus-like vesicles that are packed and sent to the apical region of the epithelium. So yeah, these data, they showed microstructure of the gills filament and cilia in high spatial resolution. And we think they evidence the production and release of the mucus-like vesicles in the ciliary cells. And we think these mechanisms can highlight the selection. We think these data can highlight the mechanism of selection and discrimination of particles to be ingested, to be further ingested or rejected by the mussel. Here we have a TM montage image of where we can see a captured food particle. So we have a set of maybe dozen images comprised a section of entirely section of the ciliated epithelius. We see here the suprabranchial cavity, and here the gills epithelium with frontal cilia here at apical region, and also a cross-section view of the lateral frontal cilia, many of them in a bent position. And also we see here what look like in auger cell covered by probably in mucus. And so we think the distinction of positioning, microstructure, and inference of functioning of the multiple types of cilia, frontal cilia, lateral frontal cilia, present in this epithelium, might shed light on the morphophysiology aspects of the gills, which interacts with the variety types of particles, including chemicals, compounds used for anti-fouling treatment in industry to combat limnopen Fortney macrofouling. And we think the outer structure characterization of this epithelium thus becoming crucial to manage the invasion process. In the next slide, we're about to finish. In the next slide, we see a closer view of the TM montage image. And interesting, we see here in a closer view the mucus that were delivered by the epithelium and sent to the region in between the filaments, the lateral frontal cilia. For example, we see some mucus, some vesicles here, and we also see some broken vesicles, and this kind of cloud that we can think is a mucus delivered by the vesicles that is interacting with the algae cell. So interestingly, we can it's tempting to compare that insect SEM image here, which showed a food particles trapped by the lateral frontal cilia, with which is almost the same size, a little bit smaller, a little bit larger actually, than the algae itself. So maybe with the size of the mucus coated particles might be almost the same size of that particles you see in the SEM image. So we think the mucus delivered by the vesicles sent to the apical region of the epithelium, they might be available for an upcoming particles that will be captured, and then the chemical interaction between the mucus and the captured particles will help the mussel to decide whether that particle will be ingested or rejected in the labial palps. So just to finish, I wanted to highlight two of researches we are currently involved. So one is from Amanda Moreira. She's a master's student. I'm co-supervising, and she's studying the morphophysiology aspects of the gills of golden mussels exposed to different chemicals. Some of them are currently used in hydroelectric power plants in Brazil. And my own research, I'm trying to understand the unresolved aspects of the mechanism of particle selection through electron microscopy. Actually, the next step will be to study the morphology in high resolution in the volume electron microscopy scope, trying to reconstruct to do a 3D reconstruction at high resolution to understand the role of those organelles in the process of particles capture. So yeah, I want to thank you for watching this presentation, and I hope you enjoyed, and if you have some questions, we can discuss that later. Thank you very much for your attention, and yeah, see you.